Greetings, all of you. Today, I'll be presenting a poster on gamma glutamyl transferase, a relatively routine enzyme which is used in the assessment of liver function tests. So, the application of gamma glutamyl tests in serum was done recently over the years to know its implication in knowing certain cerebrovascular conditions, cardiovascular conditions, neurodegenerative disorders, and certain oral conditions like periodontitis. So, periodontitis is a host mediated inflammatory disease which is mediated through the generation of reactive oxygen species. These reactive oxygen species are usually encountered by the antioxidant defense mechanism of the body. The intracellularly present gamma glutamyl transferase catalyzes the conversion of glutathione reduction which is present extracellularly. So gamma glutamyl transferase enhances the body's antioxidant capacity. So assessment of this antioxidant defense mechanism helps in knowing the antioxidant status of the body. So, the literature so far has guided me to study the salivary GGT levels in periodontitis subjects and compared them to those of periodontally healthy individuals in serum as well as in saliva to know the diagnostic ability of saliva to correlate it with the serum levels in both the case group and the control group, for which I have taken from the case group and the control group, the unstimulated whole saliva. The method of collection is by spitting method, wherein after every 60 seconds the subjects are asked to spit the collected saliva in a pre-weighted test tube which will further be weighted and centrifuged if there is any debris which was noticed. So, the serum collection was done by collection of 2 ml of venous blood from the antecubital vein and subjected it to centrifugation of 2500 rpm. Post centrifugation, serum was isolated and subjected to a semi automatic biochemical analyzer for the assessment of gamma glutamyl transferase levels. Both the serum and the saliva samples were subjected to the semi-automatic biochemical analyzer after the addition of reagents of the gamma glutamyl transferase test, which is routinely available. A digital output of all the samples was taken and the data was recorded as mean and standard deviation, following which the samples data will be subjected to the relevant statistical analysis to calculate the correlation coefficient, and depending on the R value, the diagnostic ability of saliva will further be checked upon as can it be a relevant diagnostic method for the assessment of the oxidative stress in periodontitis patients. As the current sample size is small and it is an ongoing project, the further larger sample size collection will help us in determining the diagnostic ability of the saliva. Also, it was said that the salivary diagnostic ability can only be assessed repetitive through repetitive measurements of the samples. The current study, which is ongoing, reveals a small sample size because of which the exact reliability and the diagnostic ability of the salivary GGT levels cannot be assessed, and its relevance as a biomarker in diagnosing the disease activity of periodontitis is still in question. So, future perspectives for this study would further include extensive studies with a larger sample size and a more sensitive analysis of the GGT, which can be done spectrophotometrically, or the assessment of the total antioxidant capacity, which can be done spectrophotometrically, would be a more sensitive analysis of the periodontitis, which can be a reflection of the systemic conditions where there would be an enhanced reactive oxygen species level, which usually occurs in a number of chronic inflammatory conditions which are on the rise. So, as the prevalence of non communicable chronic inflammatory conditions increases, the assessment of the oxidative stress through a non invasive method, such as through saliva diagnostics, can be a future perspective for this study, which would help in not only diagnosing periodontitis but also knowing the underlying systemic conditions which can be reflected early in a feasible, cheap, and a non invasive diagnostic method.